Have you ever wondered why fireflies glow? Well, fireflies are bioluminescent organisms. So the reason fireflies glow is because within the firefly there is a chemical reaction taking place. The product of that reaction is light. Bioluminescence refers to a chemical reaction that results in the production of light. But in nature, organisms often use this trait as a way of either a defense mechanism against a predator. Often they use it to attract a mate, so it's an attractive trait to have, and they also use it to locate food. The reaction. So what exactly is occurring that's causing the production of light? So in order for the reaction to even occur, a few things must be present. We must have oxygen, ATP, and magnesium. So here I've included an image that shows the different variations of luciferase, if you notice. And there's also different reactants. You know, we got D-luciferin here and furamazine here. And so the first person to actually give this very generic term to these reactants was Raphael Dubois. He was a French pharmacologist, and the way he deciphered and gave them their name was based on what was doing what in the reaction. So luciferase was the thing that was not being consumed, and it was speeding up the rate of the reaction. And D-luciferin was being consumed, and it was being oxidized. So you can see it here being oxidized, the production of CO2, magnesium ions are present, and we're producing light along with the oxidized luciferin. So when this occurs, when our luciferase binds to our luciferin, light is produced. In my research, we're going to focus primarily on firefly luciferase and nanoluciferase, but all of these luciferases are commonly used in scientific research. So we learned about luciferase and how it produces light and what is needed to produce light, but what role does it play in biomedical research? So luciferase systems are often paired with bioluminescent imaging, and this is just a way that researchers are able to monitor tumor growth. It's very common and it's very well known, and it's a non-invasive technique. That's why it's commonly used, and it allows researchers to test effective drug treatments. They can see if something's working effectively. So as we learned in the previous slide, luciferase needs an ATP source. So cells that are alive are that source, but when cells are dying or they're dead, more specifically, then they're no longer producing ATP, meaning that light will no longer be produced. So we can use luciferase in this technique as a whole to monitor cancer progression and also detect the number of cells that are alive based on the amount of light that is being produced. In the previous slides, we learned about the general reaction of luciferase, and we learned about the primary components that are required for the reaction to result in the production of light. Two of those being oxygen and energy. So how are researchers able to use this to monitor tumor growth? Well, what happens is that cancer cells are being altered to carrying luciferase, and the relationship between light and energy are proportional. So let's say cancer cells are dying, then the amount of light they're producing should decrease or should be non-existent, not visible, no longer visible. But if the cells are alive, then light will be produced. I've included some images here that kind of show some different characteristics of luciferase as well. So let's say like the color that's being produced. The color being produced by each luciferase varies, and a reason for that is because of the amino acid it's surrounded by. So here it's by serine, so it's producing like this yellow greenish color, and over here it's producing like a red color. And fireflies, for instance, produce a yellow color based on the arrangement of their luciferin molecules, which is our substrate. So the research I focused on was based on monitoring tumor growth and how luciferase can be effectively used to learning more about the mechanistic pathways of cancer and observing progressions, growth, and if it takes like a small number of cells, like how much cells need to be present in order for light to be produced, or for a tumor to be for tumor growth or development to be observed, and whether anti-cancer treatments are effective. So I've included 3 of the studies, and these are kind of all have the same underlying message of monitoring tumor growth and how that information can be used to better understand how tumors progress and grow. This first study was published in 2008 by researchers from UT Southwestern Medical Center. And what they actually did is they used luciferase to test whether another drug was effective. The drug they used was a vascular disrupting agent. And the reason they did this was because this agent would stop the blood supply to tumors, and the formation of new blood vessels is often the reasoning, or it results in tumor growth and spreading. So they took these mice models that were implanted with these tumors, and they were able to non-invasively monitor tumor growth. And based on their findings, when they did use this vascular disrupting agent, they did notice a decrease in signal within this breast cancer cell line that was transfected with luciferase. So it was altered to carry firefly luciferase. In the second study, researchers wanted to show how it only takes a small number of cells for tumor growth and progression to be observed. And they also highlighted the importance of these early stages of tumor cells. So what they did is they re-engineered a firefly luciferase and called it Luce2. And then they took a human breast cancer cell line and transfected it with the firefly gene. They produced these stable clones, and then amongst those clones, there was four of the brightest clones. Then they chose one, and they isolated here. It's the Luce2 1A4, and then they implanted that into different sites of the mice model. And within those different sites, they actually implanted six different locations with a different number of cells, but only 3 cells were detected. However, in another, then they went again, and then they did 5 to 10 cells into a mice, and then that actually resulted in the development of tumors. So you can imagine that, like, it only took as little as 5 cells for tumor growth to be observed. In the third study, researchers wanted to show how luciferase can be used to test the efficiency of a drug, and they did it a bit differently than the other 2 in the sense that they used dual reporter genes. So not only did they have their bioluminescent marker or reporter, but they also had a fluorescent marker, and that could be viewed with a fluorescence microscope. So they also highlighted that point that some patients have where cancer comes back, and known as the relapse point. And this is an indicator that tumor cells were actually still present, but they just weren't being detected. So the procedure they used involved the same steps as the other 2 studies, where they took a mice model, they injected it with a luciferase containing cancer tumor cell line, and then they observed it using BLI. In this case, the difference is too that they used a BCL1 lymphoma cell line, and in other 2 studies, they used a human breast cancer cell line. They separated the mice into 2 groups. One group received treatment, one group had no treatment. Of course, the group with treatment did show a decrease in tumor growth, but it did reach that relapse point. And after several weeks, the cancer had spread to different parts of the mice. And it's actually interesting that the mice appeared to be in a normal state. However, their cancer had progressed so drastically. So that's something interesting to note. This is just an image of some of their findings here. So yeah. What lies ahead for the future of bioluminescence applications? I think that this technique will continue to be a useful tool for researchers in better understanding complex diseases. I think it can possibly even be the tool responsible for finding a cure for cancer. The fact that there's already new improved variations of luciferase and substrates. So the creation of nanoluciferin, which was the most recently available luciferase, and it's smaller, more stable, emits a stronger light, brighter light, and it's currently being used to study viral diseases such as HIV. Also having new substrates such as infraluciferin, which emits red light instead of a yellow-green light. This is actually beneficial because it's more easily to be observed. So red light shines through blood and other tissues, whereas yellow-green light is absorbed by blood and tissue, so it's hard to see from within. So I think that if things continue like this, luciferase will continue to be a really good tool, a really good resource for researchers to better understanding complex diseases such as cancer.